Okay, hello everybody. Welcome to the Barnegat Bay Partnership's Ask a Barnegat Bay Scientist webinar series. Tonight we have Dr. Christine Thompson from Stockton University. She's going to tell us all about the Tuckerton Oyster Reef, which is a partnership project, and she'll tell you all the partners that are involved, but Barnegat Bay Partnership is proud to be one of those partners. Just a few things to go over. So if you move your cursor down to the bottom of the screen, you'll see a row of icons, and we're going to use the chat feature. That's the third one in from the right. So you can enter your questions, and Dr. Thompson will pause a few times, and we'll try to answer some as she goes, and she will answer your questions too during her breaks in between her PowerPoint. So I think with that, we've got 23 here and more drifting in. So Dr. Thompson, you can start whenever you want. Just real quick, I do want to mention there are some more coming up in our series. It's on the slide on the screen, and you can get the links to register for them by going to our website. So we have some good ones coming up too. Okay, thanks, Dr. Thompson.
Okay. So I'm just bear with me a second.
Sure.
Advancing for whatever reason. Try one more time. Okay, there we go. Sometimes it gets sticky and I don't know what. Okay, everybody. Thanks for coming out tonight, or for probably staying in. And thanks, Karen and the Barnegat Bay Partnership, for having me in this series. So I'm going to talk about the Tuckerton Reef project today. So this is a restoration project, and I'm going to talk a little bit about what that means. And it is a project, as Karen mentioned, that is a partnership between Stockton University, where I'm an assistant professor in the Marine Science Department at Stockton, and then Parson Seafood. This is Gail Parsons is a local oyster man that has really helped move this project along. The Jetty Rock Foundation, and our funding has primarily been through the Barnegat Bay Partnership. Again. So I have three main parts of this talk, and so there will be a chance to ask questions after each one of these sections. So first is what I call Oysters 101. So simple background biology of oysters to get you familiar or re-familiar with the oyster, because you really need to understand oysters to understand how we restore them. So the middle part, I'm going to talk about the Tuckerton Reef Restoration Project, which is in Barnegat Bay. And then I'm going to focus on the role that Stockton mainly plays, which is the science and monitoring aspect of the reef project. So part one. So I actually gave part of this talk to a middle school group, and I decided to keep some of these comments in. So this is where you can be a little interactive. And I want you to think about an oyster, because in biology and invertebrate zoology in particular, we classify organisms to their groups. So which one of these is the oyster most closely related to? So which category does it go into? Is it a crustacean, a mollusk, a fish, or a mammal? So you can just put your answer in the comments. You guys are like my students. Can you see any coming in, Karen?
Yes, and I just want to remind everyone, when you put it in the chat, please make sure it goes to all participants, and that way we can all see it. So we're getting a lot of 3 Bs and 1 A so far.
Okay. Yeah, and when I sometimes do this in live classes too, like a couple people will respond and then everyone just copies what the most common answer is, which in this case it's right. So B, a mollusk. Oysters are in the mollusk phylum, which also consists of things like the octopus, which is a pretty different animal, but they still have some basic characteristics that make them similar. But a common misconception that I find, particularly with younger students, is that they think that because they're considered shellfish, they equate them to things like crabs and lobsters and stuff, but they're just in completely different phyla. But, at least you got that. And I gave, I said that they were invertebrates as well. So C and D are both vertebrates and they don't apply. Yeah, this is sticking. So there are many species of oyster, and the oyster species that I work with, and that is pretty much the only native oyster species on the East Coast, is the eastern oyster, Chrysostrea virginica. And they're considered estuarine species, although they can withstand a pretty wide range of salinity tolerance, but not completely fresh and almost completely salty. Their native range pretty much extends the entire eastern seaboard of the United States. The Gulf of Mexico and the Caribbean is kind of its southern boundary, and then north into Canada. They are both aquaculture, so a species, so commercially grown, and also commercially harvested where there's natural populations. But as I'm going to talk about in this talk, the natural populations are kind of hard to come by. And their important role in their environment, in their ecosystem, is that they're reef building. And we can actually call them ecosystem engineers because of their ability to stack on top of each other. They grow cemented to each other, and then they can create this big three-dimensional type of structure just through their bodies and their shells interlocking with each other, creating three-dimensional habitat that also can provide special benefits to the ecosystem. So a little bit about what's inside an oyster. So many of you maybe have tried an oyster or enjoy oysters. Maybe you're eating oysters right now. Wouldn't that be fitting? But what's important to understand is that gooey blob that is inside an oyster actually has organs and structures just like we do. So here are just some of the anatomy of the oyster. So the mantle is kind of a soft piece of tissue that encases the body. The body it's between the shell and the main body. They have one main adductor muscle, which keeps the shell closed and allows it to open and close when it opens to filter. Gills are really important. These are what filter the water that comes through the oyster and brings the food that it eats, which is typically phytoplankton, into its mouth, which is shown here. This is called the visceral mass, and inside the visceral mass contains its organs, like the stomach and intestines, and they have a heart. They don't have red blood like you and me, but they will, they have what's called hemolymph, and that kind of pumps it and circulates fluid around the body. And then just a fun fact, if you ever shuck an oyster and it looks like this, and there's this big kind of mayonnaise-like blob sitting over the body, that is actually its gametes. And so during its reproductive season, which is seasonal here in New Jersey, they get very creamy like this, and that would either be eggs or sperm that it's going to release. And so that's actually my segue to talking about the life cycle of oysters, because really to restore a population or to manage a population, you really need to know about its biology and particularly its reproduction. So oysters are what we call broadcast spawners, which means that they don't have sex. They release eggs and sperm into the water, which fertilize outside of the oyster. And so upon fertilization, after about 24 to 48 hours, they develop into a larva. They have a larval stage. And so a villager larva is the name for a baby oyster. And it drifts in the water column for a few days. For oysters, it's about a couple weeks. And it grows and it feeds on phytoplankton. When they're a little bit older, so at this point, they become ready to settle. And they, at this point, the larva is known as a petty villager. But they must settle onto a hard surface in order to make that transformation to become a juvenile and then an adult. And after they settle, they can't move anymore. And so we call a settled larva a spat. And so you can see in this picture here, I'm just going to dot these. All of these little specks are baby oysters that have settled onto a shell of another oyster, which is sort of part of our restoration process. So now I'm going to talk about the benefits that oysters provide to the environment. But so in order to perform these benefits, they need to exist kind of in these dense reef clusters. So that's kind of an important prerequisite to having these environmental impacts. Oysters, they improve water quality by filtering material through their gills, and then they deposit that material into the sediments. So you can kind of think of them as like a mini Brita filter of the sea. So the metric that's kind of often quoted is that a single market-sized oyster, so about a 2-inch oyster, can filter 50 gallons of water a day. And we did this little experiment last year at a festival, back when people got together, and we put a cluster of oysters in one jar with bay water, and then just a plain jar of bay water, and this is Barnegat Bay water, in another jar. And we just waited, and after an hour or two, the water where the oysters were filtering was crystal clear, and then the control remained cloudy. So it just can kind of visually show the impacts that they can have. So if you think about, big oyster reef with hundreds of thousands of oysters can really, make an impact in water quality. When oyster reefs grow in shallower water and near shorelines, they can slow wave energy, kind of acting like speed bumps, and also keep them from eroding. So this is like a buffer for the salt marsh. It's going to keep the soil from eroding away and stabilize that. The nooks and crannies that are created by oysters growing together and stacking on top of each other will provide habitat for other species. So, some ways that species use oyster reefs: fish lay eggs in the shells, they use them to hide from predators, and then you have larger species predators that lurk around because they know that their prey is hiding within these shells as well. And then finally, at the top of the food chain, so the top predators, such as, even land-based species, such as waterfowl and humans, will consume oysters. So bringing that connection to land and to people as well. So when there was a natural oyster fishery, the fishers would take the shells out, they would shuck them, and they wouldn't get put back. They would just be discarded in these big piles, kind of shown here outside of a shucking house. So not only did that cause, the environment to lose a population, reducing reproductive output, but you also are losing that substrate that I said that the oysters need to attach to. So this was kind of the first hit that oysters face, was due to fishery. People like to eat oysters. They were harvested, but they were not harvested sustainably. And so with less oysters, less 3D structure, it also made it a lot harder for the reefs to persist, and they would get buried. And so here is what it looks like when a reef has been buried by sediment. Around the 1950s, a few diseases showed up in oyster populations, particularly, invasive or due to transplanting oysters. And it hit really hard in around the 1990s, which crippled restoration efforts, particularly in New Jersey, but other places as well. And so one of the examples of these diseases is called dermo. It is a pathogen, a parasite that can be really hard to control because it spreads rapidly in high temperatures and when oysters are in dense clusters, like in reefs. So one way that you can control for this outbreak is oysters wear masks and stay 6 feet apart. Just kidding, but that would probably work. But it's by finding natural resistant oysters and breeding them artificially to kind of create populations that have a little bit more of a chance to not die from the disease. And then you have environmental changes. And by this, I mean anything that can change the environment the oysters live in. If it's too warm, if it's too fresh, if it's too salty, that can disrupt the preferred conditions in the oyster's environment that they may have gotten used to living into. So you can think about many types of changes that can cause these changes, climate change, as well as just physical disruptions, increasing the flow through an inlet, for instance, bringing more, salty water, more rains or dams, bringing more fresh water can disrupt the conditions that it's used to. I mean, oysters are pretty hardy. Living in estuaries, conditions can change, but too much change or more consistent change can sometimes bring problems, bring disease, things like that. And then there's pollution. So this can be like toxic pollution, like heavy metals from runoff, as well as in Barnegat Bay, it's more like suspended sediment leading to murky waters, and particularly nitrogen pollution. This comes from runoff, from wastewater, and this can spur changes in the environment and the food quality, creating harmful algal blooms and kind of creating bottoms that just look murky and messy, covered in macroalgae and silt, which is not a prime environment for oysters to settle in. And so globally, this is not just a New Jersey problem, East Coast problem. Globally, there has been a 64% decline in oyster habitat. So those environmental conditions coming into play, and an 88% decline in oyster biomass over the past 100 years. And Barnegat Bay in particular would fall into that, 90% or more decline in their natural population. So I'm going to pause here because I see that there is a question already. And so if you've been thinking of a question, you can, put it in the chat. And we're also checking the Q&A as well.
So there was a question, I think Jim did answer
it.
Yeah, he answered it about the parasite, whether it moves in the water or requires a vector. That was the question.
So yeah, it can move in the water column, but I think it might need the oysters to proliferate a little bit, or at least complete its life cycle. That is a good question. I've actually been reading a lot about derma lately. Yeah, but it is kind of, it does use the oysters.
I think that's the only one I see so far.
Okay. And now it's sticking again. I don't know why it does this. Okay. I'm going to move on to our restoration project. So hopefully I have another question that I would like you guys to participate in. And I want you to think about what restoration means. And so it can be kind of any term. I put in some Google searched images that I got when I searched for the word restoration. But even though I'm going to talk about it in the ecological context, just come up with a definition for me. You can put that in the chat. I won't be grading it. Anybody care to come up with a definition?
So one person said a renewal or a facelift.
Yeah, reestablishing a thriving environment, bringing something back to its original state, reviving what is there, return to a previous state. So this is, what I was thinking you guys would kind of say, thinking of it as kind of, fixing something up or kind of going back in time, a lot of like previously back to some original state, some original, perfect condition or what it was. And, some of these pictures kind of show, but what, refurbishing a car or a house or something like that. But with oysters in Barnegat Bay, it's kind of even a step further back because it's more like rebuilding, recreating. The population is run down or completely not existing. So it's not like you're taking a framework to work with. We're kind of left with nothing. So what we want to do is bring back the oyster population, and with that, all of the services that they can provide. And you can also think of oyster restoration as just one piece of maybe the bigger picture of restoring Barnegat Bay overall. And so restoration in ecology terms, doesn't just necessarily apply to this particular habitat. It could be salt marsh restoration. It could be, rebuilding forest, rebuilding streams, emptying dams, and, recreating waterways. So there's a lot of ways that this part of science can be applied. There's a little bit of feedback. I don't know if one of the other panelists, you guys could mute, maybe that would help. All right, thank you. I don't know if everybody else was experiencing that. So now focusing on New Jersey, and particularly their oyster restoration in New Jersey, this project is not the only oyster restoration project in New Jersey. And I think there's probably people tuning into from, who work on some of these other projects. So in Delaware Bay, this is probably kind of the biggest area of oyster restoration in New Jersey. There's a project called Ports and a few others with some groups. In Northern Jersey, New York Baykeeper has a project in Raritan Bay at the Naval Weapons Station Earl. And there are a bunch of smaller projects on the in the coastal bays with the American Littoral Society and the NJDEP. They all use the same species of oyster, but the methods to recreate these oyster reefs all vary. And so here are just some examples. These are concrete blocks that are stacked together. This is sometimes called oyster castles. And then the oysters kind of set on the castles. The American Littoral Society uses mesh bags that they fill with shell as the substrate that they either set oysters onto or the oysters will attach to. And so our site is kind of located, well, in the middle of New Jersey, so it's in the middle of these projects, and in the lower part of Barnegat Bay. So this is kind of a lengthy slide, so I'm just going to give you a crash course history of the past 5 years of how we established this site and what's been done. So as I mentioned in the beginning, this is a multi-partner project with Stockton, Parson Seafood, and the Littoral Society was part of it originally. So we were given funding by the Barnegat Bay Partnership in 2015. Next year, oysters were planted at the site. We used two different types of approaches. So one was remote setting, where we put cages, and I'll talk more about this in detail. And we put shell in the cages, set the oysters on them. This is called remote setting because it's not done it's done remotely from the site. And then what we also did was we transplanted oysters from the Mullica River. I should probably point out the Mullica River is kind of right down here, just south of our site. And so that was kind of what established the reef. So in 2017, we did a second planting of remote set oysters on the reef. In 2018, we received funding for a second stage, primarily for water quality enhancement through the partnership. And with this second project, it's kind of more established partners with Parsons, as well as the Jetty Rock Foundation, and Long Beach Township plays an important role with getting us recycled shell to build the reef with. So throughout this whole, 4-year period, we've been monitoring the reef, seeing how the oysters are growing, survival. We do disease assessments. I meant to change the slide and I forgot. So this year we expanded the lease to 4 acres, and then the next planting actually happened last month. Was in June. So here's the location again in the southern part of Barnegat Bay. So the nearest inlet is the Little Egg Inlet. So this is where the reef is. So as I said, it's a 4-acre site. It's relatively small compared to the acreage of Barnegat Bay. And so what I'm showing you here is a bird's eye view of the reef. And this image was actually taken from side scan sonar, which is a machine that uses sound reflection basically to create a picture of what's on the bottom of the bay. And so things that are hard will reflect that sound differently. And so the brightly colored areas are where we place the oyster shell. And so it allows us to kind of delineate where the different reef areas are. So here we have these two strips, our 2016 and 2017 plantings, mainly where we put oysters set onto whelk shell. So I kind of call this the old reef. And then the new reef, but this is an image that was created in the spring. So it doesn't include the 2020 planting, but you can see that our last planting last year was much bigger. And so this is what we call the new reef, which was planted with recycled oyster shell. So altogether, even though it's 4 acres in total, only about half an acre is actually densely planted on the bottom. So they're kind of planted in these little strips. And it's important to keep them close together because that's what creates that habitat. So what I'm going to show now, and I tested this today and it wasn't too glitchy, but I'm going to try not to talk too much, is this is an underwater video. It was taken 2 years ago, so the oysters might look a little different now, but what they actually look like underwater. So if you were scuba diving, it's going to be that. This might be what it looks like. So it is in a good flow environment. You can see some of the macroalgae, moving with the current. And then you have the whelk shell is what we use, and then there's oysters that kind of come out and grow out. And you can kind of see they're slit a little bit, so they're filtering water. And, they're open and they're filtering. They're not all dead. And then towards the end, you kind of get a glimpse of what the regular bay bottom would look like, just with some shell hash, and not much three-dimensional structure. So I'm going to talk a little bit about the methods that we use to build the reef. So as I mentioned, this remote setting technique involves these large tanks where the shell gets caged and put into the tanks. The tanks get filled with water. Then inside the tanks, we add disease-resistant larvae to the tanks. And we get these from hatcheries, Rutgers, and there are a few other hatcheries where we have ordered oyster larvae from. And these are selectively bred to be relatively resistant to dermo, and MSX is the other one. So the larvae will then set on the surface of the shells that are in the tank, and they grow out for about a month until they're, pretty sizable and ready to be planted. So then the cages are lifted out one by one. They're dumped on a barge. The barge gets moved out to the reef site. They are planted by being hosed off of the barge. And then the dirty work is done by myself and a team of students when I could have them. So this picture was taken last year. We are way too close together for that to be allowed this year without masks. But basically we count all the individual larvae or oyster spat that have sat on the shelves to give us an idea of how many are planting. Now, this may not seem like an obvious question, but if you've worked in oyster restoration, shell can be really hard to come by. It's not readily available, and some of it is used and crushed up to make driveways. So with the help of the Jetty Rock Foundation and Long Beach Township, we created this shell recycling program. And so the main idea is we involve restaurants that are serving oysters to the public. So they save the shells from when they shuck them. They get picked up. There's it's very coordinated, a weekly schedule of picking them up. Sometimes it's bi-weekly. They get delivered to a curing site, which is basically a pile in the sun where the oysters kind of bleach off. So any kind of pathogens or germs that they have can, get cleaned. Then we go through the whole remote setting process. We plant them, and then ideally they grow, the spat grows and becomes an oyster reef. And so this is an example of a shell that somebody probably ate at a restaurant that has, 6 or 7 live oysters growing on it. So it really kind of creates this whole full circle thing with the farmers that grow the oysters for the restaurants, the people that consume them, and then put them back in the bay. So this is just my stats table, and I add to it every year. I'm just gonna, of all the plantings that we've done. So bushel set, so this basically just means amount of substrate used. So talking about recycled oyster shells, 1 bushel is 600 oyster shells. The larvae used, so this is the total number of larvae that we set in tanks. It varies sort of depending on the number of how much substrate we're using and how much, we can spend. And then, so this is the number that we get when we subsample and count all the spat to give us an estimate of the total spat that we plant on the reef. And the success ratio, so this is for the remote setting success, how we sort of document how well the oysters, survived in the tank. This is the number of spat that's set divided by the number of larvae that we put in overall. So we're really hoping for kind of a record number this year. But for those of you that are on the East Coast, when we set them in May, there was a big cold snap. And so that might have been part of the problem, that, the tanks got too cold and the heaters couldn't maintain them. And so we did kind of have our lowest percentage success yield this year. But our metric is we try to aim for 10% or above. So, even being our lowest, it is still what we aim to do. And so in total, we have almost 5 million oysters that we have planted through remote setting on this project. So right now, as I mentioned, we have the old and then the new reef. And so this is kind of, these pictures, this is last year, this might be a couple years ago, but they're growing really well. So this is, 4-year-old oysters. These are the newly planted oysters last year. So this is kind of some rough data that I just threw together yesterday. But in our last reef survey, oysters were either very low density, to only about 4 per meter square, to the new reef is very high density, so about 124 was the highest that we had. And so the sizes, so if you think of like a market size oyster, something that you would get that's about 2.5 inches. So our 4-year-old oysters are like twice as big as something you would get in a restaurant. So they're growing really big. And then our 1-year-old oysters are actually almost to market size at this point, going into their 2nd year. There's 1.5 growing seasons, essentially. All right, so this is where I can take questions. We have a question here, and anyone else?
So there's a question, do you believe the pandemic will affect the amount of recycled oyster shells that you'll have for planting this year?
Yes. Yes, because the restaurants were closed for a period of time. And so people aren't eating them at restaurants, they're not getting recycled. So there was, I think in mid-June, when restaurants were allowed to reopen, the shell recycling program started back up. But, losing Memorial Day and, those first few weeks of the summer, and people just aren't, going out as much, I don't think we'll collect as many this year. But on the flip side, there was a project that was funded by a Sea Grant to plant some overgrown oysters from farmed leases that they can't sell, because when the oyster gets too big, they're not a marketable product. So they actually might be placing some of those oysters. So we might actually benefit a little bit from the pandemic in some ways.
So the only other question, sorry, did I interrupt? Do you want to?
No, I was just going to read the question, so you could read it if you want. Or
yeah, I mean, I've been scanning them. The other one I noticed was someone asked if it's really possible to restore to some time periods, such as colonial times.
Yeah, so that is, this whole idea of, shifting baselines and what we're really trying to achieve here with these projects. And, I would say that we can't. I mean, first of all, we only have the accounts from explorers and things like that talking about, how vast the oyster reefs were. But it's not, I mean, there's a few quantitative values of stock assessments and stuff dating back to the early 19th century. 20th century, that we can go off of. But, the environment has changed a lot. And even in Barnegat Bay, like the recreational boating and stuff like that, like you can't really grow, 6-foot reefs in Barnegat Bay without, boats would, scuff themselves up and stuff like that. So, I don't really see it getting back to that. But, with all these programs, New Jersey, Maryland, New York, the more oysters that are planted, the more chance that it has to get back there. But, those reefs took hundreds of years to build. So, I don't see that, really, getting back to what it was. It's more like rehabilitating, repopulating, things like that.
Okay, you're getting a lot of questions now. So one person is asking, what is the average life cycle age of an oyster?
So the average life cycle, the age of an oyster, I mean, I think they can live up to like 15 years if, they're not fish, and they don't die from dermo. But, the reality is, in Delaware Bay, an oyster's not going to live past 2 years because, dermo's going to get it. So it's hard to say, but, yeah, I think they can be like 10 to 15 years if they keep going. I know they can live at least 4 in Barnegat Bay.
Will the Orsted wind farm project that's proposed, will that help or hinder the restoration project?
This particular, I mean, if it's the one off Atlantic City, I don't see how that would impact the oysters because they're, more inshore and the wind farm is offshore, unless there is a Barnegat Bay one that I don't know about. But I could, and, anything that like kind of creates a substrate for oyster to settle on. Like if you put a windmill in Barnegat Bay, I bet you would get oysters kind of attaching to the turbines. I don't know if that might hinder the project, the oyster project. In grad school, I did a project about wind farms and mussel farming together. So, I mean, there are some aquaculture type applications that you can get with wind farming as well. If the unmarketable oysters are used for reef building, are they still capable of reproducing naturally and self-building repopulation? They should be. There are some types of oysters that are not able to reproduce. They're triploids, so they're reared for their meat and not for the reproductive value. But so we would only be taking the oysters that have the ability to reproduce. So that's what we're hoping that they generally would. Like we know the farmed oysters in Barnegat Bay do reproduce, and they may be already contributing to our reef. So yeah, I mean, that's the idea. If they can't be sold, instead of just, chucking them, we should put them somewhere where they can, enhance the ecosystem.
Did you see there was one question where the person is asking, did you make the current reef based off of a historic reef, or did you use a sort of an algorithm to pinpoint the best area for the reef?
So where we found the reef, where we found it, so one of the sites, Good Luck Point, was based on historical reef. But in this particular area, it was created due to the bottom, that it wasn't in an area that was, too impacted by tidal flows and stuff like that. That, it was deep enough where the boats wouldn't scuff it and stuff like that. So, I mean, there was a process. We looked at the aqueous soil map and, looked at the substrate, the bottom hardness that would be best so the oysters you don't want to put them in silt or something where they're just going to sink and get covered up. So you wanted to have enough bottom hardness so they can, grow out of it. Yeah, but it wasn't really based on any kind of historical plots. And then what's the best substrate for reef restoration? I actually did a study last year where we put whelk, oyster shell, and surf clam shell in the same tank and and I looked at which ones had more. Oysters had more overall, but they did seem to settle most densely on the surf clam shell. And there was like a project in Rutgers that kind of found the same results. So it's interesting, like that smooth bottom of a surf clam shell seemed to be really attractive to the oysters, but they didn't grow very well after that. You could argue, I mean, oyster shell generally tends to be the best one that oysters kind of hone in on. But concrete can be used, it's used very often, particularly in areas where you can create more high relief structures that are deeper or maybe intertidal where you're not going to get as much boat traffic. And they can help because they kind of, bring the oysters up a little bit so they don't get buried as much, and creates more three-dimensional structure as well. But I think if you looked at like the amount of spat that's set on concrete versus an oyster shell reef, you're going to get more spat that's set on an oyster shell reef. So it all sort of depends on how you want to build it. There's no really one right way to build an oyster reef.
Okay, one more. Can seagrass beds near oyster reefs help or hinder the oyster reefs?
Yeah, so I don't know if this question is getting at maybe some of the stuff that Dr. Lacey might have talked about, but we do have a project to try this out. Hopefully next year we can start that. And the consensus is that they would help. Seagrass can contribute to clearing the water a little bit too, like they can trap sediment, kind of making and harden the bottom, making it better for oysters to grow. Oysters may benefit seagrass a little more because they're clearing the water, allowing the seagrasses to grow and come up as well too. So there is, but it's considered to be a mutualism with seagrasses and oysters nearby. They can mutually benefit each other, but there's been some studies too that have shown that oyster reefs, I don't think this would happen in Barnegat Bay, but can kind of smother seagrass beds or, make the nutrient environments in the soils not as habitable for seagrass beds. So, I mean, there has been some negative impacts as well, but I think the positives for Barnegat Bay at least would outweigh the negatives for putting them together. Yeah, so this part usually brings the most questions, is what I found.
So I just want to remind everyone to please try to address your chat questions to all panelists. That way we will all be able to see them and help answer them. I think that's it.
Okay, well, I will move on to the science and monitoring. So this is where you're all going to fall asleep. That's okay. I just wanted to point out, so these are some of my colleagues, Nate and Steve, with this is back in April, but with their COVID protocols in full force. So when we monitor the reef, there are three main objectives to the scientific part of our reef monitoring. So the main one, number one, oyster growth and survival. Are our oysters growing, surviving? Are there signs of reproduction? Number two is habitat value. So looking at the other species, does it show increased abundance of other species along the reef? And number three is kind of a new one that we've brought on board in the past year, looking at water quality benefits and how much water are these oysters capable of filtering. So the oyster monitoring surveys are done twice a year, usually May and October, although we didn't get out till June this year. We do density surveys using patent tongs, shown here, and so these are pretty fierce. They actually take a 1 meter squared section of the bottom, and so this is what it looks like when we bring up a sample. So this was part of the old reef. You can see the whelk shell. And then we sort them. We count all the live oysters. We look for spat. We take some back for disease surveys and for biomass surveys where we shuck them and we weigh them and get an idea for how much biomass, how much meat is in that oyster. And so this helps us answer this question is basically, how is the reef doing? Or, there's been some studies that have kind of come up with this threshold density, or how many oysters that you need to maintain the services that oysters can provide. And so one, it assumes market-sized oysters. And ideally, they would be, self-sustaining and producing more each year. So this was from October, the 2019 surveys. And so I haven't put in the new data as much yet. And so you can see that the old reef is kind of just barely holding on to this critical density, the structural density, which is 15 oysters per meter squared. But the new reef has more than surpassed it. And this year, the numbers were a bit lower. I think the average was in like 90 or something like that. So there is some mortality, but it's still, we're hoping that it can, you know, maintain this target for a few more years. For habitat value, so we do this in a variety of ways and kind of look at 3 different categories of habitat enhancement. So for encrusting or epifaunal species, so these are basically organisms that grow on top of the oysters. So you can see this picture here, this is actually a white crust tunicate. We have sponges that you might have seen in the video and some of the picture. So we just take like a visual assessment of what's growing on the shells. Then more quantitatively, we look for modal species that can move around, but they're more residents. So tiny crabs, worms, and snails. Basically, we put up these what we call substrate baskets. We fill them with shell. We put them on the reef. They get, you know, attracted to the shells, and then we count what comes up in the substrate baskets. We're not actually doing this study this year because it was too hard to implement with our, you know, COVID guidelines. But we are still doing fish trap surveys. And so these are to get, you know, somewhat larger species, maybe more transient species. So the ones that are, you know, going in and among the reef. And we deploy these on the reef and off the reef to see if there's a difference in fish using the reef versus off of it. And so we deploy these for about 2 days. And when we try to check them alongside the substrate baskets, but this year we're just doing it about monthly. And so this is just some of the species that we found in these surveys. I try to place fish into different categories based on whether they're reef dependent. So usually these are the smaller ones like blennies and gobies that kind of lay eggs and hide in the shells. Then there's some that are structure dependent, like black sea bass or the oyster toadfish. I mean, its name is kind of gives its association with oysters. Sometimes they're called oyster crackers. They have these teeth and jaws that can, you know, chomp down on the oysters. We find structure-oriented species like seahorse, pipefish. This striped burrfish was showing up in a lot of our samples last month, like more so. We usually get like 1 per season, and we had like 4 or 5. So he's kind of a new kind of visitor this year. And then throughout the years, we've been seeing increased number of juvenile fish. So that's really promising because it shows that the reef benefits the fish populations as a nursery habitat. You know, and potentially filling an important, you know, data gap showing structure-oriented species that aren't regularly sampled in seines and things like that. Similar to fishes, invertebrates can be reef dependent or more transient. So like mud crabs are kind of like the #1 species that show up when you build an oyster reef. They love oysters. Blue crabs aren't particularly attracted to oysters, but they'll show up to find their prey there. Spider crabs are another, and shrimp are also very much associated with structure and oysters. So I put together some of the fish trap data from 2019. So this shows the map of the reef. If you remember, this is the sonar map where you place 4 traps in and among the old reef was the only one there at this time. And then we did 4 control traps. This is not the actual site, but it's just showing that it's not on the reef. So we put them 48 to 72 hours, depending on the conditions. And so this is combined data for 3 months that we sampled. And so I had to standardize it to CPUE, which is catch per unit effort, to standardize for the different deployment dates. And so basically the average catch per trap for 4 of our most abundant species. So for all of them, there was increased abundance on the reef versus off the reef. I did run statistical tests briefly, but they weren't actually statistically significant. But you can see, particularly for some of these very reef-dependent species, that we do get, you know, more oyster toadfish and spider crabs on the reef versus off the reef. And then there's other species that make their homes around oyster reefs. So some directly use the shells for habitat protection to feed on other things like limpets and nudibranchs. Others just, are there, because everybody else is. They're like cities. So different types of species that we found. And then some of the species are problematic. And so we try to make note of them. So one of the predators of oysters are actually a relatively small snail called an oyster drill. So they use this proboscis right here to drill these pinholes into the oyster shells, and they will suck out the flesh of the oyster and kill it. Typically more of the younger oysters because the shells are a little thinner. And then they'll lay their eggs on the shells and babies will hatch, and they'll, clear out a whole, we'll sometimes get a whole cluster of oysters that have just been all drilled. A second pest, something like an ectoparasite, is a boring sponge. So here's a head of this yellow boring sponge. But what it actually does is it gets into the shells and dissolves the calcium carbonate that make the shell, weakens the shell, makes them very brittle. They can break off. They can be more easily preyed upon. So it can kind of harm the oyster that way. And then fouling species, this is more of an indirect harm. It doesn't directly harm it unless it can't open its shell if they get completely encrusted over. But these species can reduce settlement because oyster larvae can't attach to a hard surface if it's covered in, a sea squirt or a sponge. And so the final thing that I am going to talk about is water quality monitoring. And so we monitor water quality to help determine the filtration rate of oysters. And so this magic 50 gallons a day number that gets quoted a lot, and that I even quoted, what does that really mean? Can our oysters actually filter that much? How can we find that out? So in reality, the amount of water oysters can filter depends on how big they are. So that should be pretty obvious. A bigger oyster can, filter more water. It depends on the environmental conditions. So dissolved oxygen that it needs to essentially breathe, that can, if it gets too low, the oysters can't function. The ideal temperature and salinity. Filtration rate increases with temperature as temperature increases. Salinities are too low, the oysters will close their shells, as well as total suspended solids. And this is like the total amount of material that could be phytoplankton, sediment. If it's too much, it can affect it. If it's too little, they might not. So the filtration rate will increase with increased TSS, or total suspended solids. And then finally, filtration rates can just simply depend on the models and that we use to compute them. So we don't actually go in and directly measure the oyster filtration, but we use numerical models based on oysters in the labs that were treated with different temperatures and salinities. And then we can come up with a filtration rate that matches these literature models. And so in our water monitoring activities, we take profiles. So using this YSI instrument, taking things like dissolved oxygen temperature and salinity on the reef. We also have continuous loggers like this buoy here that we put out this year to get, more consistent long-term data that we swap those out, I think, every 3 weeks. And then discrete water samples. And so this is what we use to look at TSS and hopefully chlorophyll in the future, which we haven't been able to implement yet. So here's an example of how temperature impacts oyster filtration. And so this was a study that a recent Stockton graduate did over this past year. So filtration is the daily rate of water volume filtered, so meters cubed, per gram carbon, so per oyster per day. So this number here, if you do the calculations, it's about 50 gallons a day, essentially, once you correct for volume and size. The inset figure here, so this is what's actually in the model, and how temperature influences filtration rate. And so basically temperature is pretty low, below 10, but it increases pretty steadily and then kind of levels off when it gets really warm. So there is a, a linear relationship at some point, and then it, slopes off. So we can then look at the daily mean temperature at our site and then calculate the filtration rate that this average size oyster would do. And it tracks temperature. And then when the temperatures are higher, the filtration rate is higher. And when the temperatures are lower, the filtration rate drops. And the second thing is we want to be able to relate filtration and nitrogen. So they're filtering water, but they're also filtering particles that are in the water. And those particles contain nitrogen. And that nitrogen comes from things like the atmosphere, runoff, and fertilizer, a big problem in Barnegat Bay. And what happens is it fuels algae growth. And the oysters eat that algae. And so the nitrogen that's in the algae then become into the oyster. And then if the oyster, defecates that material, that nitrogen is then in the sediment. And so that can either, be buried, it can also be transported. And then there's these microbial processes, particularly denitrification. So the nitrogen can transform its forms from ammonium to nitrate. And then the denitrifying microbes can actually take the nitrate and convert it to nitrogen gas and get it out of the system. So it goes back into the atmosphere. So the more oysters you have filtering water, clearing the water, the more potential nitrogen that you can have removed as well. So not only they're clearing the water, but they're actually taking out something that could potentially be harmful. And so this looks like clearance rate, which is the amount of particles that an oyster can clear from the water. So we take that filtration rate and then we look at how much it can clear. And so we're using chlorophyll as our proxy for nitrogen at this point. And so the idea is that higher chlorophyll is more food for the oyster, and the clearance rates are higher. And so we have, clearance rates tracking with the chlorophyll trends and the chlorophyll spikes. However, so this is who's really paying attention. Down here, all of a sudden, it doesn't quite track. So you have a spike in chlorophyll, and you don't have the same type of spike in the clearance rate. So what could potentially be happening here? Does anybody have any ideas? Nobody? It's that temperature. Anybody?
Yeah, one person said, one person said data error, and another person said too cold. And
okay, I'm not seeing these for some reason. Maybe they're not going to everybody.
Some people are sending the chats to just the host, and so I'm the only one that can see them.
Okay.
And then one person said, did the oysters die? A major weather event. They're coming in fast now. Reduced grazing in colder weather, colder water.
Yeah, so it's the temperature dependence on the oyster clearance. So yeah, so when, I can't go back because my computer's freezing again. Yeah, if you go back to the temperature dependence, in October through November, the temperature falls 10, 15 to 10, back to where, it's the temperature effect is going to not allow the oysters. And eventually they go to about 0. So, the chlorophyll can increase all at once and the oysters just, go dormant and they don't filter anymore. So, particularly a seasonal area like New Jersey, the bulk of the filtration and the clearance is only going to happen in the summer months. And so that's pretty much all I have. The idea, the take-home points here is, restoration everywhere is an evolving process. And, we have plenty of challenges, and our challenges might be different than other challenges elsewhere. But the best we can do is keep collecting the data, and, monitoring things and, adding to the reef and hopefully starting to see more improvements. We're definitely seeing that with, the fish and the species that live there. And, You know, it's a multifaceted, multi-partnership endeavor. This is not something that Stockton can just do. It's not something the Barnegat Bay Partnership can do on their own. We really needed the, Dale's help with his, remote setting facility. Rutgers has, their shellfish lab that has, done the disease tests for us and stuff like that. So it's really been a project that just keeps growing. And so, hopefully our site can expand as well. We're hoping to expand to creating another reef next year. So I can take any last questions now. And I just want to thank my collaborators, my co-PI, Steve and Anna, and Dave, Elizabeth, and Nate, who are basically the five of them are kind of running this program right now because I'm more remote than I typically am this summer. And then all the students, these are just the students that helped them pass year. So it's, fun when there's not COVID to get students out and helping and stuff like that. It's a really, cool experience for them to be part of this as well. All right, I think there were a couple questions. I saw things about cow-nose rays. Did they get answered?
Yes, Jim answered that question. There was one asking why, see if I can find it. When she shucks an oyster, she sometimes finds small crabs in them.
Yeah, so I've not actually seen one of those little pea crabs in any of the oysters that I've shucked on this project. But yeah, they sometimes called oyster crabs. They're technically, I think, parasites, but I think some of the studies haven't really shown a negative effect. They kind of live inside the oyster and grow in there, and use it for the ultimate protection. Where is the potential new reef site? It is actually not permitted or anything yet, but our idea is going to be around the Route 72 bridge, Manahawkin area. So a little north of our site, but not as far north as the Good Luck Point site, which was one of the original sites that we planted on, but the oysters didn't grow as well there. And there's also going to be another site. The American Mineral Society has a project at Forked River where they're going to plant oysters probably next year as well. That's a little further north.
Somebody did ask a question back to the wind farm again, asking if the cables from the wind farm, if they're scheduled to be laid from the sea to Oyster Creek Power Plant, would the magnetic lines of flux generated by high power affect the oysters in the bay? I
don't know, but I wouldn't mind getting funding to investigate that.
And someone asked, do you do your own permitting?
Technically, the short answer is yes, but it's not. Steve Everett actually goes through the paperwork process of that. But yes, we do. Stockton does apply for the permits through the NJDEP, and the Army Corps, I think, has to approve them as well.
I think that's, let me just check the Q&A real quick. I think that's it for the questions.
Really good questions. So pretty much cut this to an hour.
I know, you did it. So thank you very much, Dr. Thompson. It was excellent. I know everyone enjoyed it. I see lots of thank yous and great presentation comments.
There's one more question from Mark. I'm just
one more question.
This. So because I like larvae. Is monitoring for larvae in the water or natural spat fall part of their other programs? So I have taken samples for larvae in the water, but there's so many bivalve larvae in there, it's really hard to find the oysters. I kind of gave up. But we do see natural set on particularly the newer oysters that aren't as fouled. And then we do put spat bags out periodically. We haven't for a few years, but then this year they started again. So, and the substrate baskets that are filled with shell, we do get oysters that set in the shell in the substrate baskets too. So we don't have a formal spat fall monitoring on the reef, but I think they are assessing that this year to get a better handle. And we have some previous data too where there wasn't a lot. So if what we're seeing this year is a lot more, then maybe we could say that, something's happening potentially, that, oysters are recruiting to this site. Can we really pinpoint if it's are oysters that are reproducing, not really, but you know, any oyster is a good oyster for the reef. So
Great. All right. So you have a student looking forward to
yeah, well,
talking with
he
you.
can sit out on one lecture, I guess, if I end up doing it.
So thanks
Ahead of
again.
the game.
That was great. And I also want to thank Dr. Jim Ladies, who was a panelist on this, helping to answer the questions. So both Jim and I work for Barnegat Bay Partnership. So thanks again, Dr. Thompson, and join us for our next webinar in the series, which will be Dr. Doug Zemeckis from Rutgers Cooperative Extension talking about the fisheries of Barnegat Bay. Thanks again.
Okay.
I'm going to end the show.